All right, let's take a look at the skeletal and smooth muscle. We'll begin with a very brief overview of skeletal muscle anatomy. I assume that most of you have already had an anatomy class and you're comfortable and familiar with this terminology. And therefore, I'm going to focus specifically on the anatomical terms you need to be comfortable with when dealing with physiology. And if you're struggling a little bit with this, you might spend some time on this figure to review some of the terminology. We'll start actually looking at the individual muscle cell, which as you look here in this particular diagram, if we can pull a little cell, what we really have with the skeletal muscles is bundles and bundles and more bundles. Each of these red cells here is a muscle fiber bundled together as shown. We have connective tissue not shown here, so we also have red blood cells, blood vessels running throughout the muscle fiber or muscle tissue to provide the oxygen and nutrients that the muscle needs for contraction, as well as to remove carbon dioxide. If we look at the muscle fiber and we pull it out and we peel back the sarcolemma, we can actually see the fibers inside there. Terminology that you need to be comfortable with: the term sarco is used over and over again when we're talking about muscle and the different parts of muscle. This includes smooth, skeletal, of course, which is what we're looking at right now, and even cardiac. So this terminology is found throughout each of these muscle types. The first phrase that you need to be comfortable with is sarcolemma. That refers to this plasma membrane. You can see we've pulled it back here in this little section here, but this entire area right here, this part here is my plasma membrane, and we refer to it as the sarcolemma. You might recall that the cell membrane goes by plasma membrane as well as plasma lemma, and that's where the term comes from. Then we have our sarcoplasm. That's this area shown here in tan. I'll go ahead and highlight it in yellow so you can kind of see which area I'm talking about, this cream colored area. And that sarcoplasm is consistent with, it is the cytoplasm, but because we're dealing with skeletal muscle, we call it the sarcoplasm. Notice, of course, that our muscle fibers are multinucleated. You should already know this. These are long cells, and they also contain, not shown, but a rich supply of endoplasmic reticulum, and we're going to talk about their function there. Let's look at the filaments more closely. So if we take a look at these filaments, these myofilaments is what we refer to them. Those myofilaments are actin, myosin, and a few other proteins that we'll look at. The myofilaments are composed with the cytoskeletal proteins, and they are arranged in a very specific order in skeletal muscle. We also see this arrangement in cardiac muscle, but not in smooth muscle. So let me jot that down. Seen in skeletal, cardiac, but not smooth. Okay, so keep that in mind. Smooth muscles don't have sarcomeres. They do have actin and myosin, but they're not arranged as we see here. So a sarcomere is a bundle of this myosin and actin, and specifically it's referred to as this unit from the two lines that I'm highlighting. These lines are what we refer to as the Z line, also called the Z disc. And so you can see the region between one Z line and the next, that's my sarcomere. And then we can subdivide that sarcomere down into two regions. From, let me kind of draw a little bit of a dotted line here. You'll notice that the Z disc runs right through a lighter section, and that lighter section is referred to as the I band. These are the light bands. I'm kind of coloring it a little bit in green. Hopefully you can see that. Of course, green might not be the best choice if you're red-green colorblind, sorry. Maybe, I don't know, yellow, but that's not good. There we go, yellow. And then you see the darker bands with a light band in between. So this light band, focus, let me kind of highlight that as well. We'll just outline that one in yellow. This light band right here, I'm going to talk a little bit more about this band later. This is the H band. And then the dark band that runs from here to, well, you can't really see in here, it would be probably right here. This region right there is the darker region here, and it does include the H band, but it's this darker region. This whole area is called the A band. Taking a closer look then, digging into that muscle fiber so we can see each of the structures. In the sarcolemma, there are these pits, and these pits are really quite important in terms of function. These little pits right here, they're invaginations, and they're not actually pits, they're tubes. But from the outside, they might look like pits. And you can see there these openings in the plasma membrane. And then if you peel back the plasma membrane and take a close look inside, you're going to notice what these pits do is they form these tunnels, or these tubes. And these tubes run throughout the muscle fiber, and kind of scribbling in there in terms of yellow, so you can see these tubes here. What these are is the T-tubules, also known as the transverse tubules, transfer across, transverse crossing. And so these transverse tubules carry, what their job is if the sarcoplasm, sarcolemma, sorry, is depolarized. So let's imagine we have an electrical signal generated in this segment of the sarcoplasm, and then what it's going to do is it's going to spread throughout that the sarcolemma. Sorry, I misspoke again. It's going to spread throughout that sarcolemma and travel all the way down the sarcolemma. But in addition to that, it's also going to travel, and this is important, it's going to travel through the T-tubules, down the T-tubules, and across to the other side. And this is critical in that it's going to carry the electrical signal inside into the cell. Remember, these skeletal muscle fibers are quite large, and they to get the muscle fibers in the interior of the cell to contract, we need that electrical signal. The T-tubules carry it through. Notice right next to the T-tubules we have these structures right here that I'm going to kind of outline in blue here for you. They're this blue-green structure that you can see here. And what these here are is these are called the terminal cisternae. If you think about cistern is a storage. It's used to store usually water, right, or fluid of some sort. And in this case, the terminal cisternae is a storage area, and what it's storing is calcium ions. We call it sequestering. We say the calcium is sequestered here. It's drawn out of the sarcoplasm and tucked into that terminal cisternae. And what this terminal cisternae is made out of, or what it is it's basically the Sarcoplasmic Reticulum, abbreviated SR for short. And so it's a swollen area, or a storage area of the sarcoplasmic reticulum. Now you're going to notice that we have two of these terminal cisternae right next to the T-tubules. Again, that's going to be functionally important. What the electrical signal is going to do is trigger the release of the calcium ions that are tucked away in that terminal cisternae. And so we need those calcium ions for muscle contraction. Notice the fibers, the endoplasmic reticulum or sarcoplasmic reticulum has many branches that runs throughout the cell, and again this allows for more surface area and greater release of calcium, and for that matter greater ability to sequester the calcium as well. The T-tubules and the terminal cisternae make up together what we call the triad, and they're critical in terms of muscle contraction, as I mentioned. Now let's take a closer look at the sarcomeres, and then we're going to look at the muscle fibers, the actual filaments themselves. Here's the sarcomere. I've already given you this A band, which is essentially from here to here, but note that we have this little band, lighter area in here. So the entire A band is this whole segment. Okay, so this whole segment that I'm outlining in blue, this is my A band, all of it. But if we go and look at the neighboring A band, so starting right here and here, you'll see that lighter area in here. That's my H zone. And I want you to focus and pay attention to the H zone. We're going to talk about in terms of muscle contraction, how that H zone changes. Then of course I have my I band from here to here, this whole thing being my I band, this little structure right there. And so that's important as well. We're going to talk about that too. And then these dark lines, you'll see this is labeled right here, but this is my Z line or Z disc right here. I just wrote over the top of the Z, so we'll just write it back in. Looking over here, let's see what we looked at the top. This is an electromicrograph. If we look down here, this is a model of the muscle fiber. And then these green lines here, these are what we call our myosin filaments, also known as our thick filaments. Not shown, but still present, there is a point of attachment that essentially runs down the middle of this layer, or this portion of thick filaments, and we call that the M line. And you'll hear me talk about the M line from time to time. Notice that, let's look at the purple fibers. These are my actin filaments, and they're also called the thin filaments. And those thin filaments, I want you to look at how they overlap. From here to here, we have overlap of those filaments. So the A band starts here, ends here, okay? So this is the beginning of the A band going all the way across this direction. But this area where there's little overlap of the actin filaments, let me kind of highlight it like so. This is my H zone. So you'll see the M line runs right through that H zone. Here's my Z discs. Contained within the Z discs, also known as Z lines, remember, is a protein called titin. It's not shown in this figure, but titin is an elastic fiber that helps the muscle return to its, it has this elastic spring to it, it generates force, and it helps it return to its resting state when the muscle contraction ends. Looking more closely, then we're zooming in here again, we have my actin fibers here. Notice the little beads, I'll talk a little bit about that in a second. This part right here is what actually makes the M line. It's this thicker region here where the myosin filaments anchor to one another, and then those filaments will stretch out. Each of these filaments has what is known as a myosin head on them. And here's my little myosin head. We're going to look at that in a little bit more close, more detail in a minute. And so those are my myosin heads. And again, just as a reminder, from Z disc to Z disc is my sarcomere. One of the theories regarding muscle contraction, and here we're going to focus primarily on anatomical changes using what is known as the sliding filament model, or theory. It's a fairly well-established theory. There's lots of evidence supporting this idea, and it's accepted as being the way that muscles contract. So let's look at the electromyography. If we see the top version there as being the muscle in the relaxed state. So notice relaxed refers to this one right here, this section of muscle right here. And we can see what we're looking at is two sarcomeres from Z disc to Z disc. So we've got sarcomere 1 over here, and we've got sarcomere 2 over here. And we can look and compare these sarcomeres in relationship to relaxed state, partially contracted, and fully contracted states. Look at this H line right here. I want you to pay close attention to that, to your A band and mostly to your H band. The H band, as we begin to contract, let's do some comparison here. You'll notice that my H band is significantly shorter in both of these sarcomeres. It's decreased in size. Go back up here and look at what the H band is. H band is the point at which actin and myosin don't overlap, so no overlap. Okay? So what we're really doing is we're shrinking the part of the muscle that has just the myosin in it. And so as we do that, what we're doing then is we're essentially pulling. We contract when the muscle fibers move. What they're doing is the actin is actually being pulled toward that M line. Okay, we're tugging on the actin and pulling it inward to the M line, and so the actin is sliding over the myosin, and hence the sliding filament theory. And it does this as myosin grabs hold of the actin and tugs it. Now, when we look at a fully contracted state, where did the H band go? It's supposed to be right here, right? But we've essentially lost that H band, and the reason is that the actin has been pulled so tightly closed that there's not a region without overlap. It's all overlapping. So overlapping actin and myosin filaments. If we look down here at the model, we can actually see this in more detail. Again, here's my H zone, the green fibers representing the myosin, the purple fibers representing the actin. And we see as we contract that the H zone gets shorter and shorter until it's pretty much non-existent there. And so that's essentially the sliding filament theory. And you can see the contraction there. Now, if we look more closely, again, as I mentioned, we're going to look at that mechanism more closely. What we're really seeing, and by the way, this is the Titan that I mentioned earlier. It's the elastic fiber that's going to help to pull things back into their resting state. Here's the myosin heads, and what they do is they actually attach to the actin, and then the myosin heads move. We'll look more closely at this. We call this the cross-bridge cycle, and I'm going to teach you that cycle in detail. And so as we have thousands of little heads grabbing on to the actin and tugging it towards the M line, then we get movement of that actin, the sliding towards that M line, which is right here, and so forth. This is entirely calcium dependent. We'll look at the exact mechanism. Remember that calcium is stored in the sarcoplasmic reticulum until we need it. Once it's released, we get this rapid contraction of the muscle and contraction. Here's a closer look at the thin and thick filaments. This is the myosin head. A single myosin fiber essentially has this myosin head, and then these filaments follow it along. So here's my myosin, you get the idea. But it forms a complex with other myosin, and so we see just here we've got two of them twining together. But now imagine up here hundreds of them twining together to form a very strong, thick fiber with literally hundreds of little myosin heads. This is the head. And we're going to take a look at that head in more detail. What I want you to know about this head is that this head has multiple functions. First off, it has an actin binding site. And that's this site right here on the top here that's going to attach to my actin fibers. Then it also has a site that binds ATP. And just as important is it's an ATPase. What this means is it's an enzyme, and it's going to hydrolyze ATP. So ATP will be broken down to produce ADP, an inorganic phosphate. And that ADP and phosphate temporarily occupy that ATP binding site. We're going to see these changes and look at how important they are in terms of muscle contraction right now. Again, I'm really kind of focusing more on structure with just a little bit of function thrown in. And then here's the last anatomical slide that we're going to be looking at. If we take a close look at those actin fibers, remember up here, this is myosin. Make that really crystal clear. Myosin, thick filaments. Looking at this one here, this is my actin fibers. And what we see here is these balls here. This little structure here is known as G for globular actin. But then these G actins, they polymerize. You should be comfortable with that word, polymer, meaning to make a longer structure from small units. And so they polymerize, and the polymerized form gives us what we call F-actin, our fiber. And it's the actin that we see in as the actin filament. Now, importantly, actin, if we look at actin, if I were to look at a single actin, here's my little ball here, what I'm going to see here is that actin has a myosin binding site. And that's good because myosin has an actin binding site. So we need a place for these two proteins to link together, and that's what we find. Now, in addition to the actin, the purple balls that we see, we are also going to see some other structures here. This pink-like structure looks like a rope, okay? This is called my tropomyosin, okay? And then these yellow structures here, this is a complex of proteins. Shown here is 3 proteins, and we call it the troponin complex, but I actually just call it troponin for short. I don't actually include the complex in the whole nomenclature. And in that troponin, so actin has a binding site. You can see troponin has a binding site for tropomyosin, but it also, one of these subunits has a binding site for calcium. So this is my calcium binding site that I'm kind of coloring in there. In the absence of calcium, it looks like the arrangement I have here, where we've just got this tropomyosin. I guess this will work. We'll color pink with pink. My tropomyosin fibers. And notice how it's twining itself around those actin filaments. And what it's going to do is a little bit of competitive inhibition. So when the muscle is at rest, what this means is that tropomyosin competes very successfully with myosin for actin binding. And so the tropomyosin blocks and prevents actin from binding almost completely, but not quite completely. The fact that we do have some myosin bound is what gives us our muscle tone, more commonly referred to just as muscle tone, which is a state of slight contraction that holds tension in our muscles and essentially allows for contraction to occur more completely. We'll look at how all of these play together in one of the next videos.